Right. First of all, we try to understand what is the meaning of gene expression. We know most of the things. Only thing is we have to bring everything to this place to understand what is the meaning of gene expression, right? Let's see what we have learned before. We have learned protein synthesis. We have learned uh, DNA replication. Many lessons we have learned. If we try to bring all these lessons, for example, protein synthesis. We have learned protein synthesis before. So let's start briefly proteins to learn about protein synthesis, or just to revise about protein synthesis. Protein synthesis takes place in the structure of ribosome. So this is a ribosome, and what we have learned, messenger RNA molecule. So this is the mRNA molecule. When mRNA molecule and ribosome both are assembled, then the tRNA molecules bring amino acid at a time to attach to developing a polypeptide chain. So if this is the tRNA molecule, and tRNA molecule has amino acid attached to it. So this is the amino acid. And this is a tRNA molecule. What will happen eventually, the messenger RNA molecule has codes. This code consists of codon, and this codon represent one amino acid. So after all, these codes read by the tRNA molecule, and it produced a polypeptide chain like this. So this is the polypeptide chain. And after that, we have learned in another lesson, this polypeptide chain is further modified and also folding. After that, we can find the correct protein molecules like this. So this is the protein molecule. And this stage is called translation. Translation. And this is the protein, right? Okay, now we are coming to the nucleus. So this is a nucleus. We are now going backward. So this messenger RNA molecule arriving from the nucleus. So to start, we have learned again the DNA molecule unwind, and the gene is converted to a messenger RNA. This is the messenger RNA, and this is a transcription. Okay, now we try to link the process. So this is the number one, transcription, and then messenger RNA is leaving to the cytoplasm, translation, and the protein molecule form. After modification, protein molecule form. So number one, transcription. Number two, translation. Then the protein folding and modification, it form a protein. So if we try to show arrows with this one, so gene is copied to messenger RNA, Then the messenger RNA is leaving from the nucleus into the ribosome, cytoplasm. And then this is converted to polypeptide chain. Polypeptide converted into a protein. Now we can call this entire process gene expression. Gene expression. So starting from the gene and ending up making a protein is called gene expression, right? That is the gene expression. So we're going to learn gene expression and regulation in this lesson, right? Coming back again, gene expression. If we want to define gene expression, so gene expression is process of producing protein from the gene is called gene expression. Okay, can you write immediately under the topic gene expression, process of producing protein from the gene is called gene expression. Process of producing protein from the gene is called gene expression.
Okay, sir.
Right. Now we are coming to a next point. We have learned previously the stem cell. Now what we're going to learn, how stem cell can change to this kind of a cell, or how stem cell can change into this kind of a cell. Now why this is important? Because we have learned totipotent stem cells, pluripotent stem cells, and multipotent stem cells. So during this process of development of the organism, stem cells change to different type of cells. After that, these differentiated cells remain in the body permanently as this kind of a cells. Example, muscle cells. So what you see here, muscle cells. Muscle cells remain as muscle cells. So if we think this kind of a cells are white blood cell, so white blood cells remain as white blood cells. So they are not changing after that. Now we're going to understand how these cells permanently change to a specialized cells, and how specialized cells remain as a specialized cells without changing to other cells. Only stem cells can change to the cells, but once it differentiate, it permanently coming to this stage, and it doesn't change after that. So if you take a human body, we can find 217 different cell types. So these cell types are originated from stem cells, and they remain as those specialized cells. It doesn't change after that. But interestingly, if you take the genes, let's say this is the nucleus of the muscle cell, and this is a nucleus of white blood cells. Then if we check genes, genes are same. So if this cell has 25,000 genes, this cell also has 25,000 genes. And then number is same and the types are same. So they are exactly the same. In our body, like one person body, his genes are same in all cells. Gene numbers same, types are same. So they don't change. But what change? The type of cells. This type change and this type change to muscle cells. Genes are same, number and types are same, but the cells are different. So now we can understand during the process of changing from stem cell to white blood cells or changing from stem cell to muscle cells, something is going to happen. Now we're going to learn what exactly happening during this process. Okay, coming again to understand the genes of a cell, let's assume this represent all the genes. So this represent all the genes of a cell. Now when these genes in all the all cells of the body, it has same number of genes and exactly the type same. But during this differentiation, what is going to happen? A set of genes remain as active. Another set of genes remains as inactive. So one type of genes going to permanently inactive and another type of genes permanently active. So in biology, we try to use this term switch on. This is switch off. So active genes are called switch on, inactive genes are called switch off. So this is the method used by the differentiation stage. Set of genes switched on and set of genes switched off. So we're going to learn this process a little bit detailed now, how this switch on and switch off thing work. Right. we are coming again to the important points to note down. So this is an important thing we're going to note down, right? So let's look one by one. What is this process in detail? Okay, this represent a cell, and this represent nucleus of the cells. And this one going to represent the DNA of the cells. Now we're going to write here process of differential gene expression. Process of differential gene expression. And cell differentiation. That means the number of genes activated genes express, we learned gene expression, express gene produce proteins, and the protein can change the cell. Okay, so we start one by one. What is the starting process? The starting process, number one. Number one is there is a chemical stimulation. So let's generalize this chemical stimulation. Now this chemical stimulation happening mostly from outside. If there is a relevant chemicals, so this chemical bind to the cells. So then if this is a receptor, and then the chemical bind to the receptor. So this is the chemical. Right? So I just write it here, receptor, and this is the chemical. Now this directly is going to activate the DNA. So it's giving a signal to the DNA molecule. So we just leave like this signaling event. So we are not going to learn at the moment how this signaling event pass into the DNA, but one chemical activate another chemical, another chemical, another chemical, just like a series of chemical activate. After activation, what is going to happen? Some genes are activated. So that means some gene switch on. So if these genes switch on, that would be the second one then, right? So second one, some genes switched on. If they switch on, they convert into a messenger RNA, mRNA. So this is a transcription. So let's give this to the next number. So this should be a third one then. So third one. Transcription. After transcription, the messenger RNA produce proteins. So this is the process of translation then. Now, proteins are active compound, active molecules. So these proteins give some kind of a change to the cells. So this is the fifth one. Protein make permanent change to the cell. Protein make permanent change to the cells. Now, this is a permanent event. Then the cell remain as permanent cells. After that, nothing changed. So only this gene switch on. Rest of the genes, switch off. Rest of the genes switched off. The switch on gene, it changed the cells. So 1, 2, 3, 4, 5. So what we learn after chemical signal transcription translation happen, protein change the cell. Okay, can you copy this diagram under the gene expression?
Okay, sir.